Hello everyone. Microbes everywhere, but not a microbe we can see with our naked eye. So we have been talking about the most ubiquitous microbes, the microbes which are present everywhere around us, but we cannot see them with our naked eyes. Talking about the ubiquity of the microbes, they are present in the oceans, they are present in the air, they are present associated with plants, they are present in the soil, they are present in your homes, they are present in you, they are present on you. So the microbes, the bacteria, they are present everywhere and they are the most ubiquitous in nature. And therefore it signifies the importance that we cannot ignore these microbes. As we will divulge into the different roles these microbes play, we will understand that how important it is to understand the microbiology as well as to become literate, the microbiology literate. Now I distinguish between the two terms, which I have one of them which I have been using quite often, the microbiome, and the another term which is termed as microbiota. Now microbiota is the assemblage of microorganisms present in a defined environment. More specifically, the microbiota varies according to its surrounding environment. The term microbiota is preceded by the name of the environment in which it is located. For example, the gut microbiota. You must have heard of the term a lot these days. It refers to the microbiota in the intestinal tract. So when we talk about the microbiota, we talk about the sum total of the organisms, the sum totals of the microbes we are talking about. So the number of microbes, the diversity of microbes, the abundance of microbes, but we are talking about the assemblage of these microorganisms, and it is usually present in a defined environment. So when we talk about the gut microbiota, we talk about the skin microbiota, we talk about the environmental microbiota, in which we can talk about the marine microbiota, we can talk about the river microbiota, and even in river microbiota we can talk about the sediment microbiota. So when we talk about all these things, soil microbial communities or soil microbial biota, whenever we talk of microbiota, we talk about the organisms, the sum total of the organisms. But as I told you, that there are limitations and there are challenges to identify the microorganisms, and with the advent of the technology such as genomics, metagenomics, metabolomics, then more and more non-culturable microbes were identified. And this expanded our understanding of the microbiota as we identify or as we limit ourselves to the identification of the microbes. And this expansion included not just the organisms but also the theatre of activities that it performs, and that is termed as the microbiome. So microbiome is referred to the collection of the genes harbored by the microorganisms and the so-called the theatre of activities. In other words, the term microbiome describes the collection of genomes and genes from the members of a microbiota in a specific habitat. So microbiota is the organisms. When we talk about in that particular environment, we talk about the entire set of genes and genomes that are present. From that genomes, that when we sequence these genomes or when we sequence the metagenome, the total DNA from these specific habitats, we can reconstruct the genomes of the non-culturable microbes. And they will also tell us that, we cannot identify them with the culturable techniques, but they are present there. And as I earlier said, that has expanded our knowledge of the microbial diversity also. So we nowadays use a lot of time the word microbiome. That is basically the genomic constitution along with the microbiota, which reflects the microbiota only, but not those microbiota which can be identified with the conventional or the culturable techniques. So I hope you understand this, and the next slide further reflects this better, because it is very important to understand the microbiome, what it means. So as it clearly reflects here, the microbiome is the sum total of the microbiota. The microbiota, which we understand in the terms of bacteria, fungi, algae, protists, and different, other organisms which are present, plus the theatre of activities that it does. Now, this theatre of activities, how we can assess them is generally by doing the sequencing at the genomic level. Then we do the biochemical analysis also, then we do the physiological analysis also, or we do the metabolomics, which is basically trying to identify the metabolism, the metabolic pathways which are present based upon the analysis of the genomes, or based on the analysis of as this omics is expanding into transcriptomics, as this omics is expanding into proteomics. So not just the genomics, but the transcriptomics, the proteomics, they all reflect to the different theatre of activities that this microbiota is performing in a set of a specific environment. So you can see that the environmental conditions is also constituted there, because with the difference in environmental conditions, the microbiome can vary. So the conditions are also present, and then the theatre of activities that we are putting it through. So the analysis of the genes, analysis of the RNA, analysis of the proteins, and they give rise to different, activities which are present, what kind of molecules they are forming, what kind of interactions that is occurring between them, what kind of metabolic pathways are there, what kind of protein-protein interactions are there. All these kind of analysis are included in the term microbiome. So microbiome is a very, very widespread term which is used to understand this microbiology, or this microbial diversity and microbial abundance in the environment, or as I would say, the microbial sphere, the microbe sphere, to understand it better. So much importance was given to this microbiome that it was decided to celebrate 27th June as the World Microbiome Day. Now this World Microbiome Day celebrates all the things microbial all around the world. So this microbes, their ubiquity has also provided a unity amongst the entire world, so that the entire world celebrate all the things which are microbial. So not only the microbes, but all the things, which means the biochemistry, the physiology, the all the different kind of activities that these microbes are performing, all the challenges these microbes are imposing, all the different kind of problems or these microbial world is also causing or creating, then all these things are basically understood. The people are made aware, and all these activities are basically recognized on this particular day, particularly which is the World Microbiome Day. When I am talking about the microbiome, I cannot leave behind the human microbiome. Now this is an introductory lecture, so I am just touching upon different things which I will be elaborating much more later in the series. So human microbiome, when we talk about, so you can see and well appreciate the fact that on the left hand side there are certain facts which are put down in according to the human microbiome. This has been taken from the World Microbiome site only. So these are the facts which are relating to us, telling us about the human microbiome, and on the right hand side you can see the different kind of microbiota which is associated with the humans. So they are associated with the brain, they are associated with the gut, they are associated with the lung, they are associated with the urinogenital system, they are associated with the skin, and you can well see the different kinds of bacteria. These are just some examples, but this is just to again reflect upon the fact that different kinds of bacteria, the microbial diversity can differ, but since it is present in a same human body, they are all interconnected. So they are all interconnected, and some particularly interesting facts about microbiome is also reflected, which states that 95% of the body constitutes this microbes. So these microbes which are present, they are even more than the number of cells in the humans. 1.3 times the number of cells which are present there. So there are around, so many microbes that are present. In fact, what they say is that when we talk about the role these microbes play, we will understand that the balance of the microbiome A perfect balance of the microbiome is very important for the homeostasis, and we will see that in many diseased states this microbiome is deviated from its normal, or as we will say, the healthy sense. These microbiomes, they are associated with the viruses also. So there is 5:1 ratio of viruses that are present. So you can well imagine again that what our body is made up of. They are made up of cells, or they are made up of microbes in fact. And these microbes, when we will understand, that is, they are governing the entire physiology of the body. We will talk about the different diseases that are caused by the dysbiosis in this microbe or disruption of this normal microbiome. We will look into much more detail of this human microbiome, and we will talk more about it. So you can again, emphasizing on the fact that we ourselves are constituted of the microbes that are present, and the functions that they perform is what is regulating our normal body which is there. So this was generally as an introductory thing to talk about what are microbes, what is microbial abundance, what is microbial diversity, what is microbial, the microbiome, what we talk about, how the scientists across the globe have understood the importance of these microbiome or the microbes, and they are basically, launched programs such as the Earth Microbiome Project, where they are analyzing the microbial communities in different biomes across the globe, so that they have the better understanding of probably the forms and the behaviors that have developed over the evolutionary scale. They understand the descent of life, and they also understand the maintenance and propagation of this life in the biosphere. So we have talked about that. Now I will take up individually each component of these microbes and make you understand upon their characteristics, about their classification, about their importance, about their habitat and niches. So that is, how we will go about them. So first of all, we will talk about the most abundant one, which is the bacteria. I take the most abundant one as the bacteria because I am taking the microorganisms as the living organisms, but I will certainly talk about the viruses in detail also. I will also touch upon the viroids, the prions, all these acellular entities, what they are, what they constitute, what are their importance, and how do they relate to humans or the human environment. What is their role? What is their significance? So I will try to touch upon all these different things in our subsequent lecture. To start with, we talk about bacteria. Now bacteria, the size of the bacteria is on the order of a few micrometers. And that is why we say they are microbes, because they are micrometer. 10 raised to the power of minus 6 meter is the size of these bacteria. And this illustration very well illustrate where the microbes lie, or where the bacteria basically lie. So you can well imagine it's a relative description of the sizes. You can see that is one strand of the hair on the top panel, and you can see the rock, the grain, the sand grain which is there, and beyond, further as we go down, it becomes invisible to our naked eye, and that constitutes the microorganisms. You can well see where the coronavirus is lying, the size of the coronavirus, the size of the Zika virus. The viruses are smaller than even the bacteria. Then certain archaea, we will talk all about the different things, but right now I want to appreciate you the fact that even the RBC is a little bigger than or it's in the range of the size of a bacteria. Now this is the average size of bacteria which we are talking about. It ranges from the 0.2 to 2 micrometers, but then average it is in the range of the micrometers we are talking about. So this illustration very well tells us about the relative size of the bacteria which is present all around us. We can't see it, but it is present. We are very well aware of the coronavirus. So you can see how small it is. We cannot see it, but we have very much felt the pervasiveness of this virus, and we will talk in detail about that in our subsequent lectures. Shape and arrangement. So coming to the shape and arrangement, there is a whole variety of shapes in which this bacteria exists. Arrangement I talk because when I initially said that they might not be occurring as the individual cells, but they appear or they exist in terms of colonies. So in the colonies, there is a specific arrangement that can be present. So some of the common shapes are basically the cocci, where they are circular in shape, or the bacilli, where they are termed because of that, where they are rod in shaped, or they can be spirally. They can be, different kinds of shapes which are present. So they exist in many number of shapes which are there, and they can further associate. For example, it is shown the diplococci, where two cells are coming together. When the division occurs, they do not separate. There are tetracoccus arrangements and different kinds of arrangements which are there. Bacilli, the spore forming bacteria. So we'll talk again about some of the division aspects and some of the physiological aspect of the bacteria too. But right now I'm just talking about the shape and the arrangement of these different bacterial species. So size is very small, ranging in the micrometers. Shape very much diverse, and very many different arrangements can exist. Looking at the phylogeny, looking at the classification, the taxonomic classification, because as I said, an important aspect is to understand about the evolutionary scale on which they exist, evolutionary relationships between them. Then this particular figure tells us about the different phyla the bacteria contain. These phyla or this arrangement is based upon the evolutionary significances or the phylogeny. Now this phylogeny, will talk about tools and techniques also when I will elaborate upon the various genomics techniques or taxonomic techniques in microbiology methods. Then this phylogeny can be assumed by the similarities that exist in the basis of morphology, in the basis of biochemical features, in the basis of physiology, but more recently in the terms of genomics. So the genomic sequences, how much they are relatable. So such arrangement, such clustering as it is termed, can be done into these phyla based upon the genomic similarities. So the genomic similarities in particularly to 16S, or as we say, entire set of nucleotide identities, entire set of amino acid identities, all these kind of analysis are done, and the clustering is done so as to put the similar microbes together. Now, as I told you about the identification of the microbial diversity, the culturable methods are little difficult because all the bacteria are not culturable. Similarly, taxonomy based only on the culture methods is also not very precise, because many bacteria which are similar in many functions or morphology or culturally, or physiologically, they may be belonging to very different clades. It can be there based upon their genome sequencing. So there has to be a clubbing of all the methods so as to ascertain the phylogeny. Now looking at this phylogeny, the most abundant is the actinobacteria is there, the phyla actinobacteria is there, which is then followed by the gamma proteobacteria or bacteroidetes which are there. So you can see the different phyla which are basically present in the bacteria. Some of the major clades you can see, some of the major phyla you can see, which constitutes majority of the bacteria which are there, but there are many other rare phyla which are also present. Because they contain so many different species which are distinct from these phyla, which is so many things, so many classification can be done of the bacteria based on the phylogeny which is there. Now classification can also be done on the basis on the cell membrane. Now this is a very, very simple thing. In fact, in the phylogenetic methods also we can divide the bacteria into different forms, and these different forms can be based upon the cell membrane. So the most important is the gram positive and the gram negative. It was a staining method that was discovered by Hans Christian Gram somewhere in like 1880s. It was discovered, and it still holds its place also, although it is not a very perfect method, because many of the even the age of the growing bacterial cells or different environmental conditions can give rise to some ambiguous results. But this is a very preliminary method which is used to classify the bacteria based on the cell membrane which they have. Now the gram-positive bacteria have got a thick peptidoglycan layer. However, the gram-negative bacteria do not have this, a very thin peptidoglycan layer is there, but they have a polysaccharide layer which is present above it. And because of that, as it is showing in the flow diagram, this staining technique is followed. So in the staining technique, the bacteria are stained with the crystal violet and they retain the color indigo color, then they are decolorized with alcohol. Now gram-positive bacteria, because they have got a very thick peptidoglycan layer, do not lose this crystal violet color. However, the gram-negative bacteria, because we do this decolorization, the lipopolysaccharide layer get dissolves into it and the crystal violet color, the indigo color is basically the stain is lost. And then we counter stain it with another stain which is called as safranin, and therefore the gram negative appears to be pink in color. However, the gram positive appears to be purple in color. So this gives us a quite distinction, and we classify different bacteria based upon whether they are gram positive or whether they are gram negative. Obviously, the gram positive will have similar kind of a biochemical and physiological properties, and gram negative have different. Although this is, again, I am saying this is just a dichotomy we are talking about. So every organism, every bacteria cannot fit into this just two things. There are certain exceptions also, and then this is not a 100% perfect method, as some spurious results can also come up. But this is a very prevalent method to classify the bacteria. Now this flowchart again tells you the different bacteria which are gram positive, which are gram negative. It also talks about the different shapes of bacteria, and it also mentions the phyla of bacteria, which can, as well as the examples of bacteria which are gram positive as well as gram negative. As I said, there are certain exceptions, and these exceptions can be when they cannot be isolated from the host cells. For example, chlamydia. It's difficult to stain chlamydia because they are intracellular bacteria. You cannot stain them easily. Then mycoplasma, we will talk a little bit about these mycoplasma because they lack a cell wall only. And then spirochetes, for example, because they do not uptake they cannot be detected under the light microscopy. Now the technique is basically using the light microscopy to distinguish between the different colored bacteria, but spirochetes cannot be identified under a light microscopy. However, need a higher microscope such as electron microscopy to be detected. So these are certain exceptions which we cannot classify, but many of the easy classification can be done based upon the gram positive and the gram negative bacteria by performing this gram staining method. Then apart from the staining methods, or apart from the cell membranes, the bacteria can also be divided based upon the nutrition. Now when we talk about the nutrition, it is basically that bacteria, they are the living organisms. They need to grow, they need to divide, and they usually divide asexually. We will talk about quite similar to what sexual reproduction in higher organisms is. There are certain phenomena that occurs in bacteria. We'll talk about them. The horizontal gene transfer methods, or certain methods such as the transduction or the conjugation, which causes the genetic recombination to occur. Quite typical like what happens in the higher eukaryotic organism, sexual reproduction. But the most prevalent method of reproduction in bacteria is asexual reproduction, which is generally by, say, binary fission. Now this is basically the growth of bacteria is with the increase in the number of bacteria. So for the growing, they require nutrition, they require energy source, and based upon where this energy source is coming from, you can see on your slide the different types of the bacteria that they are classified into. So they are proto-autotrophs and they are heteroautotrophs. Then heterotrophs can also be saprophytic, they can be parasitic, they can be commensals. We know that E. coli lives in our gut. When we talk about the gut bacteria, at least one of the bacteria you are very well aware of, the E. coli, which stays in our gut bacteria, it is a commensal bacteria. So there are saprophytic bacteria, there are commensal bacteria, but there are harmful bacteria also, which are the parasitic bacteria which are there. So they do not prepare their own food, but the proto-autotrophic, where they are basically preparing their own food, they can be sulfur reducing bacteria. So what kind of source they are utilizing? First of all, whether their source is basically the light, or their source is chemical energy. If it is light, then it is the photoautotrophs, and if it is the chemical energy, it is basically talking about the heterotrophs that we are talking about. So you can well understand that depending upon what energy source they are using, oxygen is used by most of the bacteria, but there are other bacteria which use the sulfur as the source, nitrogen as the source, or different chemical elements as the source, and therefore there are different types of bacteria that can be found. So this classification is based on the nutrition. So classification can be based on the cell membrane, the classification can be based on the evolutionary time scale or the phylogenetic relationships, the classification can be based on nutrition, and as you can appreciate the fact that these bacteria are the most abundant, most ubiquitous, present everywhere, including in you and on you. So I stop here and we discuss further in our next lecture. Thank you.